Namaste Vidya, Hamisha Khushro Vidya. A very good morning to you all. Myself, JV'n Dr. Ravi Jain, Assistant Professor, Department of Practice of Medicine at Faculty of Homeopathic Science at JAYOTI VIDYAPEETH Women's University. In today's lecture, we are going to discuss about the genetic factors. Genes are the functional units that encoded in the double-stranded deoxyribonucleic acid or the DNA that are packaged as chromosomes, and these are located in the nucleus of the cell. The genes are the functional element of the chromosome that are capable of transmitting information from the DNA template via the production of messenger ribonucleic acid or the mRNA to the production of proteins. Gene activity or the expression is influenced by a number of complex interacting factors, including the accessibility of the gene, promoter to transcription factors. Now, there are two types of cell division. Cell division takes place by two processes. One is the mitosis and the other one is the meiosis. Mitosis is the resulting in two diploid daughter cells, and meiosis is the resulting in the four haploid daughter cells. Stages of cell division. The cells are not committed to mitosis are said to be in G0 stage. The cells committed to mitosis must undergo through preparatory phase of the interphase that is consisting of G1S and G2 phase. G1S and G2. So we have G0, G1S and G2. The G1, the stages of cell division. Now, in the first stage, G1, there is the synthesis of cellular components that are necessary to complete the cell division. S phase is the synthesis phase, where the DNA replication producing identical copies of each chromosome that is called the sister chromatids. G2 is the second gap, the repair of any error in the replicated DNA before proceeding into mitosis. Mitosis M phase consists of four phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromosomes condense and become visible. The centriole moves to the opposite ends of the cell and the nuclear membrane disappears. In the metaphase, the centrioles complete their migration to the opposite ends of the cell and the chromosomes, consisting of two identical sister chromatids, line up at the equator of the cells. In anaphase, we have the spindle fibers that attach to the chromosome and pull the sister chromatids apart. In the telophase, the chromosome condenses, the nuclear membrane reforms, and the two daughter cells, each with 46 chromosomes, are formed. Meiosis consists of four phases, the same: prophase, metaphase, anaphase, and telophase. But it differs from the mitosis in the following ways. It consists of two separate cell divisions that are known as meiosis 1 and meiosis 2. It reduces the chromosome number from the diploid to the haploid number via the tetraploid stage. The reduction to the haploid numbers occurs at the end of meiosis 2. Constitutional genetic diseases. The familial genetic diseases is caused by constitutional mutations which are inherited through the germline. The pattern of disease inheritance consists of autosomal dominant inheritance, autosomal recessive inheritance, and X-linked inheritance. So we have three types of disease inheritance: autosomal dominant, autosomal recessive, and X-linked inheritance. Now we also have another term, that is somatic genetic disease. Somatic mutations are not inherited, but instead occur during the post-zygotic mitotic cell divisions at any point from embryonic development to the late adult life. The most important example of human disease caused by somatic mutations is cancer. So this was all about today's lecture. In the next lecture, we will be going to start with the genetic disorders. This session was powered by Digital Version 2.0, JAYOTI VIDYAPEETH Women's University. I hope you are satisfied with my digital session. If you have any query, please mention in the comment box and I'll try to resolve. So this was all for today. Thank you very much. We will be meeting in the next lecture with genetic disorders. Thank you.